We get experimental. SciAnts. We're curious, non-judgmental.
I wanted your perspective on this because this is also, I guess, a type of genetic modification. They're infecting mosquitoes with a bacteria called Wolbachia, and that is decreasing the incidence of dengue in Australia.
Okay.
And they're releasing them in a controlled manner to where if males that have Wolbachia mate with females that don't, those females become sterile. The females that do have Wolbachia have a massively decreased likelihood of passing a dengue onto a person. Is that same flavor of GMO? Like, is that a GMO because you're just infecting it with a bacteria or not?
The genome of the Wolbachia has been changed?
No. No genome of anything has been changed.
Then it's not genetically modified.
Because
it's biocontrol.
Some of the popular news articles are saying because they weren't normally infected by this, then they infected the mosquitoes with Wolbachia.
Sure.
And that's a form of modification.
We call that biocontrol. Genetic modification requires the modification of one's genetics at a minimum.
Yeah, I just wanted your take on this because there was a similar kind of vibe to some of the popular media articles of like, oh, this is like in some way modified and it's bad.
So is the Wolbachia not endemic to the area?
No, it's not. So
Okay, so it is like biocontrol, which is always something that's worth talking about. Like we're releasing something into the environment that is not normally here. Like humans are making a decision to release this thing, and we've seen how like invasive species can cause problems, and so that needs to be done carefully and thoughtfully. But anybody who'd call this genetically modified anything, I worry has not actually read the paper, and therefore I would suggest probably should not be reporting on it. That's my take though.
There are genetically modified mosquitoes. Yep, in the Keys. Like, we've talked about those in the past. This one just came out, I think, today, this paper. And again, it's this bacteria called Wolbachia. We'll talk about it in a second. But they infect the mosquitoes with it, and that even if the mosquito is infected with dengue, it can't pass it on to a human. And they are not sterile, Sparky. These are not sterile. It's just if a male with Wolbachia mates with a female that doesn't have it, that female will actually become sterile.
But the ones in the Keys that are sterile that they release, they have to continuously release those. Is that correct?
That is correct. Yeah, I just wasn't sure which one Sparky
I wasn't sure either, so I thought I'd get out in front of it.
Male system makes the female sterile. Yes, that is correct. For the ones that are released in the Keys.
Yeah.
Here, if the males are infected and they mate with an uninfected female, that female's sterile. But they're releasing, Sparky, not just males for this experiment. There's males and females infected with the Wolbachia because the goal isn't to necessarily wipe them out. It's to make it so that the likelihood that they pass on dengue just plummets way, way down. So I just wanted your quick opinion, just because like there were some news articles too where they're like, oh, this is like a genetic modification and it's like a terrible thing. And it's like, I also didn't think it doesn't count, like, because it's not genetically modified, but is it get into the same argument because they are introducing a non-native bacteria into the animals and spreading it?
I mean, they're completely different considerations. Is it something that we should think about very thoughtfully? Yeah. We're taking something that is not native and putting it into an ecosystem. We've done it before. We don't always make the right choice. We thought, why not just add this species? Yeah, the ecosystem will be fine, right? So yeah, it's absolutely worth a very thoughtful conversation. I think the conversation is entirely different than genetically modified crops and other things used for food sources. Like, one is being cultivated, and it's very different, and certainly it's not genetically modified, and I'm concerned for those journalists, but it's certainly worth a thoughtful conversation about it. I don't want to be dismissive if people are like, I have concerns. It's like, well, we probably should. It doesn't always go well. Doesn't mean we shouldn't do it ever.
And no, Sparky, nothing else will get it. Like, it's not good. If something like a Wolbachia bacteria is not going to infect you or a mammal.
Right.
It's it like other insects, sure, but the mosquitoes won't be eating a fruit fly or injecting something into the fruit fly to allow it to spread. So it won't have the same effect, no. So again, I thought this was a tangentially connected article because they do talk about taking care of mosquitoes, keeping them at bay through the use of modification. What's wild too about this, y'all, like in lab settings, Wolbachia, so like it seems like the usual insect lifespan, let's say for a fruit fly, is 2 weeks in the wild. Wolbachia won't cause any damage for 2 weeks, but if you infect your fruit flies with Wolbachia in the lab and then you age them, they start rapidly getting holes in their brains, which then leads to memory, learning impairments, a bunch of, like, as you would imagine, neurological issues. And so almost all the papers that you'll see that are looking at behavior, the flies don't have Wolbachia, and the strains of the lab strains don't have Wolbachia, because they can influence behavior, and if you're doing sequencing experiments on the brains of the animal, like, there's just a lot of variables that they introduce. However, there are some lines that you can get that do have Wolbachia, and I know we had them in the lab isolated away from the others just to make sure they couldn't cross-pollinate it anyway. I mean, it would already be like contamination of your stuff, but if you got contaminated by one that's carrying bacteria, would be even worse. And so in order to avoid that, they were, sequestered off into a different part of the lab, which was interesting. That's the level of, like, almost, I guess, protection that we had on them, right? Is that this paper that just came out had been testing it in the field. Like, they've already been releasing, and they were doing field collections to see what the status of the dengue infections are, and to see how efficient and efficacious this infection approach actually is. And it seems like it's very good. For the Wolbachia, either, Ian, it's a different strain of Wolbachia than what is naturally in the population, or no Wolbachia. So both of those scenarios lead to this cytoplasmic incompatibility, where two cell types are coming together, but one is infected and one isn't. They're, like, at a crossroads, and it just, like, doesn't end up working. Like, the embryo is not able to undergo the appropriate types of divisions. Like, there's just, it's at the very beginning, there's a stopping point. Right here, they're using it in a place where these mosquitoes are invasive. This isn't the same approach what's being done in Australia. Here, they're just releasing infected mosquitoes with Wolbachia. They're not wiping out the population because there's animals that depend on them as food sources. So the reason why we don't just wipe out all mosquitoes is because it would be, like, it could cause an ecosystem collapse. So when locations where they are endemic, instead of killing all the mosquitoes, you just infect them with this Wolbachia, and then they're unable to pass on the dengue when they're infected. So I think it's a neat intro to the idea of Wolbachia and how we are able to use it as a potential form of biocontrol. I've got this one here: introduction of Aedes aegypti mosquitoes carrying this particular strain of Wolbachia in Australia. The graphical abstract, pretty straightforward, right? Dengue incidence goes down with Wolbachia frequency going up. Here, they don't want to decrease the population. They're just trying to decrease the incidence of dengue. And so it's given the location of, like, and I guess what's the purpose, it's neat to see how it's adaptable for that Wolbachia infection. So Wolbachia, y'all, the big takeaway, does a lot of different things. And what I think is really cool and can be frustrating is that because it does so many things, people actually will actively ask, if you're submitting a paper with insects and you're looking at behavior, you're looking at any of these features that he's talking about, one of the first reviewer questions is, does it have Wolbachia? Oh, no. So, Dino Dave, it's not that they won't read the paper, it was a collaborator, right? So if the collaborator was going to work with us on behavior stuff, he was very big on if the insects have Wolbachia. And so first question was always, did you test your insects and make sure they don't have Wolbachia? And so you don't put it anywhere on the paper, you don't need to put that. It's just, if you see a weird behavior that you're not necessarily expecting, that your normal lab lines maybe are predicted to have behavior X, and you start seeing why, a question a reviewer may have is, well, did you test for Wolbachia? Are your stocks infected? See, that's what we were talking about for, like, the brains of the fruit flies as well. When they get old, and we saw that in the lab too, as they age, it's just, like, totally trashing the brain of an animal. And that is something that these researchers in the study we're chatting about now had to, is that they did in fact have to inject embryos with the Wolbachia to grow up lines that they can then release in the wild. Wouldn't necessarily stop them from mating and reproducing and having an increased population size, but again, it would stop them from spreading dengue. This setup is kind of how Lito was saying too, of, like, how important the public buy-in is, right? Of, like, going and demonstrating the opportunity and the reason for releasing something like this. Get rid of bodies altogether, you will crash the ecosystem, Dino Dave. Mosquitoes do a huge amount. There's over 1,000 species. Huge amount for pollination and food sources across many, but blah, blah, blah. Then you destroy the world, Dinosaur Dave. Particular approaches, there is the possibility, right, that maybe if these animals, gotten rid of, that there would be a collapse in the food chain. It's probably getting rid of 1 out of 1,000 is the likelihood of that is pretty low, relatively speaking, right? Now then, we do have a little bit about dengue here, so I thought it'd be a neat way of talking about just like what's the important, like why is dengue not a good thing? Just a little bit of an introduction of what exactly dengue does. Yeah, fortified pink without public buy-in, never gonna come through. Never ever gonna happen. Especially as something like releasing a genetically modified, or not even genetically modified organism, right? As Lita was saying, they're just infecting them with a bacteria, but you're still releasing them a bacteria in your yard that, again, like if in a mosquito that might also end up biting you, and it's just further issues that can form, right? And the difficulty of pulling folks in. Yeah, it could be right dead on arrival, no matter how good the science is. It could be a very big issue if you don't have that public buy-in. And at least, you know, if you can demonstrate and make the argument, it's not that it's not beneficial, but like there's this disease state, right, that you could be able to help eradicate, that might further facilitate and get people to actually do the buy-in. Probably still not easy. I mean, Ian mentioned in the Keys where they release these modified mosquitoes, not in a Wolbachia sense, but actually genetically modified, there's a very difficult flavor of buy-in there as well. So right, there's a difficulty. If you've been infected by one flavor or one strain of the dengue, getting infected by another one doesn't mean you're actually protected from via the first infection, but rather there's an increased likelihood that you'll have a much worse reaction. So it's this unique type of additive infectional state. Michael, I know, it's a say what. Point, Michael R. Hassler, is that it's bad. And if we have the opportunity to fight it, I think it would be a good thing to do, right? The reason I showed this video is just the importance of why we should care about dengue in general and why it would be a good thing to be able to get rid of it. Right, like you said, it's scary and it's not good. Like it's, you know, even if you survive the first one, the likelihood that you'll get infected by a different strain is pretty high, and the doublet of it actually makes it even worse in the second round of infection. So if we can get rid of it at its source by not having it be passed on to you via mosquito, that would be great. The way you can do that again is by infecting the mosquitoes with Wolbachia, and then the likelihood of them passing on. I mean, there are studies that suggest that Zika also would be as likely to be passed on, but it's definitely been shown in dengue. They're not actually injecting them into the adult, they're injecting into the developing embryo, and actually they're injecting it in a very particular time, pre-syncytial stage, meaning before the embryo goes from like this multinucleated state in a single cell to having single nuclei per individual cell. That's the particular stage at which they are doing the injections, and that's a big catch. So again, localized regions of the city, they release some mosquitoes that have Wolbachia, their other region doesn't have Wolbachia released, and you see how many dengue cases they are, and 318 to 67, huge decrease in the amount of patients that do present with dengue. So it is clearly a successful appearing method to get rid of the dengue in the city. And that's key, right? Because there's those 4 different strains, and those 4 different strains, when differentially infecting you, might do well with the infection of one, but infect multiply, that's when it becomes worse. And so this kind of goes into Fortified Pink Snake's comments, is that they're releasing Wolbachia infected mosquitoes into the city, and the other city regions, like neighborhoods, requested that there be more released as well in their areas as well. I mean, that's the sign of a very good public buy-in, right? So this is a nice, very different approach than what we were chatting about with actually getting rid of all mosquitoes. Here, you're just making it that the host that is the mosquito is not a good place for the virus to grow. And so you're essentially making it not a habitable location, so the mosquitoes still survive. They're still being eaten by everything else, and the food chain isn't interrupted. They can still pollinate. They can still survive. Not interrupt the ecosystem, but then the diseases aren't spread further either. So it's almost a win-win. And I want to wrap up tonight with this ethical question that I think we kind of answered in using this approach. And the ethical question is just if we should be getting rid of all mosquitoes in general. This approach here does not get rid of all mosquitoes, right? We maintain the mosquito population. They just have Wolbachia, so it's different. But just again, something to think about is, should we, if we had the opportunity, get rid of mosquitoes? Which is, again, this is where the video is coming from, of like the dilemmas of releasing something like this, which is why I like the idea of the Wolbachia. Now, they haven't tested if the malaria, because I mean, that's not a virus, that's a parasite that's living inside the mosquito. The question would be, does it work there or not? And so I don't know if they've actually tested it in that particular disease state, but it would be an interesting one to test there. How low would the level have to go? For what, KK? Like how much the total population would have to be decreased by? To drop the malaria? Oof. That's a hard one, because I think like they'll build a bunch of these models to predict like how the population might spread, but there's so many like external factors on population spreads and those approaches that we just don't know what's gonna work and what's not gonna work. Could the bottleneck recover? They will just all die. I'm not sure, KK. The two examples that they gave when they've done this, they led to a massive population collapse. And so it's just the question of would that end up happening or not? Yeah, that's, I think, the nice way of using Wolbachia, right? Because that doesn't lead to population collapse. That just prevents the mosquitoes from passing on diseases. By diseases, they've looked at Zika and Dengue closely. They haven't looked at malaria. But imagine if it did work with using malaria, then maybe it would be a better reason to use that, and then you're not going to collapse the population. So that is, y'all, I think the summary of this paper here. So they introduced Aedes aegypti mosquitoes carrying this particular strain of Wolbachia, decreasing the incidence of dengue in disease hotspots. And this was done in Australia without plummeting the population of the mosquitoes. So they're still part of a food chain, anything like that. It doesn't matter, like that's maintained, that's not being affected. They're just not passing on dengue. The mosquitoes don't die, exactly, which is, again, maybe KK, you could argue, well, it's just being paranoid because why not just go ahead and kill them? But I don't know what's going to happen if we do kill them all, right? Like you were saying, if like collapsing those species. So it's, I think, best be more mindful, not just wipe everything out indiscriminately, but being aware that it could have long-term effects. And this is the great way to stay on top of that.